Hello, good morning. This is Peter Nagy, Chief Medical Officer of Praxis Genomics. Thank you for the organizers for allowing me to speak. I'm going to talk about constitutional genetic disease diagnosis for the 2020s based on the combination of methodologies that I think will be crucial for making progress in sensitivity and specificity of clinical diagnosis. So a little bit about Praxis Genomics. We are a CLIA and CAP accredited laboratory. We do comprehensive genetic testing and counseling services, and we use Bionano Saphyr system as a centerpiece of our offerings. So our mission statement is really to bring diagnosis to the 50% of people who do not get a diagnosis even after doing extensive cytogenetic microarray panel and exome and maybe even genome sequencing. And the way we plan to do that, or we are doing it, is combining optical mapping data with Illumina short read based whole genome sequence data, and finally transcriptome sequencing to do functional assessment of the changes that we find in the genome. So the services that we provide, again, are clinical genetic consulting. So we helped our customers to choose the proper test. We do the proper testing itself, the clinical genetic testing. And we also do research projects when arranged separately. So this is a summary slide in a way. It kind of illustrates what the current paradigm is for genetic testing and what our company is offering. So you see a general odyssey, diagnostic odyssey, starts with potentially karyotyping, then a microarray, then a single gene sequencing. Maybe you choose another gene and you sequence that. Then you go to maybe panels. Then you go to exome, then a new exome comes out from a different company, then you repeat the exome. 2 years go by, you still see that the exome has expanded, and you order another exome. If that comes out negative, then you maybe start to think about other options, maybe mitochondrial genome sequencing if you didn't do that before, or you do mitochondrial depletion testing, or you do repeat expansion testing, or maybe you test for repeat contraction testing, like for FSHD, if your patient symptoms fit into that category. So instead of all these tests being performed sequentially, or maybe with some overlap, what we are offering, just start out right away with Bionano Saphyr optical mapping, a whole genome sequencing, and analyze the two data sets together. And then whatever you find, verify it in a transcriptome. If the gene or the finding of interest is expressed in blood, then you can use a blood transcriptome. If not, you use the appropriate tissue if available. So the technologies that we use are summarized on this slide. You see, of course, the Bionano instrument and the Illumina NovaSeq. And then we use Genox to do combined analysis of the two data sets. And we do transcriptome using Illumina sequencer and the HiSet suite to analyze the data. Currently, Praxis Genomics is the only company that offers a clinically validated Bionano testing in combination with whole genome sequencing. So our validation had three main components. The first one, we validated the sample types we can use. So currently we accept fresh blood, frozen tissue, and cell lines for our analysis. Secondly, we established our reference range and reportable range and the performance characteristics of the Bionano system. And thirdly, we did a list of samples with different types of mutations, representative of the different types of disorders that we will be asked to diagnose. So this is our diagnostic test offering. You see Saphyr optical mapping, proband only, duo, trio, additional family members. We also do offer it as a carrier test in case other carrier tests fail to detect the cause of repeated pregnancy loss and infertility and so on. We, of course, offer whole genome sequencing and also whole genome data analysis if the sequencing has already been done somewhere else previously. The combination of the two tests, the Saphyr optical mapping and the whole genome sequencing. And in addition to that, we offer FSHD testing using the Bionano Saphyr and also repeat expansion sizing using the Bionano technology. So the output of all these tests that we perform, the genome analysis and the SOM analysis, is actually provided in a single report. So that single report, even though the data for it comes from many different analytic pipelines, it's still going to be an easy to interpret and brief report, basically containing the variants or the changes that we truly believe are the causes of the patient's problem. So I would like to illustrate how Bionano performs for the different types of variants that I listed above in the validation. So I want to show you how the FSHD testing actually presents itself. This is a slide summarizing the pathogenesis of FSHD. It's quite complicated, but the most important concept here is that one has to size the D4Z4 repeat both on chromosome 4 and on chromosome 10, and one has to establish whether the repeat on chromosome 4 has a polyadenylation sequence or not. So that pretty much establishes whether the patient has FSHD type 1. In addition, testing for SMCHD1 deletions or point mutations will complement the picture. And also, we do additional methyltransferase assessment to make sure that we catch the FSHD2 cases. So this is sort of what Bionano can do so very well. It can size the repeats, the D4Z4 repeat, like on the top of the slide. You see, that purple repeat has a repeat count of 4, and below that you see a larger repeat. So that larger repeat has 28 units in it, and you see both the short and the long has the 4QA allele, which means that it's polyadenylated. So from these two, only the top one will cause disease. Then you notice the third one from the top, the third picture from the top, illustrates what happens when the repeat has a non-polyadenylatable allele, and that's the 4QB allele, the similar repeat size. And finally, you see how the technology can tell apart the chromosome 10 allele from the chromosome 4 allele. So you see the yellow lines with the little blue dots here are the actual reads, and you see the pattern between the differences in the pattern between the 4QA and the 4QB alleles that are surrounded with a red square. And also you see the difference in the pattern between the chromosome 4 and chromosome 10 on the left side of the screen. So the top three images show chromosome 4, and the bottom image shows chromosome 10. You can see that the gray bars have a different pattern. And so that's how the system tells the size and the origin of the different variants. So the next type of very useful application of this technology is the repeat sizing. So repeat expansions are very common cause of disorders, underdiagnosed. They can be expensive, and until recently, generally they were offered as a single test, one by one. So you sort of went through the whole list. The nice thing about BioNano is that it can actually provide a sizing for those repeats that are large repeats and before could only be sized by southern blots. So oftentimes the sizing is very critical, and it influences, the presentation and the severity of the disorder. One example is DM1, and this is how a DM1 case will show up on BioNano. So you see the size of the region, including the DMPK gene. The normal fragment size there, the BioNano fragment size, based on the labels that are deposited, is 19,363. And then you see the expanded allele, where the same markers are 24,422 base pair apart. And then so you can calculate the difference, and that divided by 3 in this case will give you the size of the repeat. So no other technology can really give it to you in such a nice, clean way. Similarly, you can size other large repeats like Fragile X syndrome associated repeats, the same idea. In this case, you can then confirm it with a transcriptome assessment, where you can see the blue arrow pointing to missing expression in the sample with the mutation that I showed above, with the expansion that I showed above. BioNano is also very useful for detection of translocations, reciprocal translocations that don't change copy number. And this is how the data shows up. It's really satisfying and quite beautiful, actually. You see the actual sample in the center aligning partially to reference 2, which is actually meaning chromosome 2, and a chromosome 21, indicating that there is a new junction established between the broken fragments of the 2 chromosomes. BioNano is also quite useful in assessing the copy number changes or structural variants in regions that are very difficult to assess using short read sequencing. And one good example for is congenital adrenal hyperplasia caused by mutations in the CYP21A2 gene. Here you can see a homozygous deletion affecting both alleles in a patient with this condition. So I think I got to the end of my short presentation. I hope I managed to give you a sense of what this technology can do in the clinical use. Just to summarize our modus operandi, we propose, instead of the stepwise single test testing that is currently practiced in many clinics, we propose a 3-component testing to be done immediately upon presentation of the patient. And that would be a Saphyr optical mapping test, a whole genome sequencing using the short read technology, and then for verification of the functional significance of these findings, that could be single nucleotide variants, small indels, copy number variants, repeat expansions, and so on. You would want to do a transcriptome test to to provide you with the actual functional significance of the changes that you find. This is actually very important when you are at the borderline between a variant of unknown significance with pathogenic characteristics versus a likely pathogenic variant, or you would be even willing to call something straight out pathogenic versus a likely pathogenic report. So thank you for your attention. And if you have any questions, please, you can contact me directly. Thank you very much. Bye-bye.